Hello everyone, welcome to the medicine channel. I am Dr. Guna here. Kindly pause to read the disclaimer fully. Let's start the gene series. Topic: Recombinant DNA technology. rDNA technology or cloning is a form of genetic engineering. It involves amplification of desired DNA sequences. Initial step involves extraction of DNA containing the desired sequence. Let's take insulin gene as an example for desired sequence to clone. DNA is cleaved by restriction enzyme EcoR1. EcoR1 cleaves to yield the desired DNA sequence insulin gene. It is a type 2 restriction endonuclease that creates sticky ends. It identifies a GATTC palindromic sequence and cleaves between G and A. Other restriction enzymes are EcoR5, BAMH1, CLA1, Hi3, HIN3, NOT1 and SIT1, all named after bacteria. Bacillus amyloliquefaciens H1, Caryophyllon latum 1, Haemophilus aegyptius 3, Haemophilus influenza D3, Nocardia otitidis caviarum and Providentia stewarti 1. Selection of the restriction enzyme depends on the sequence it cuts. Then prokaryotic bacterial cells like Escherichia coli are taken. They contain the extra chromosomal circular DNA material that is capable of autonomous self-replication called plasmids. Example PBR322. Plasmid Bolivar Rodriguez 322 in E. coli also have genes that confers antibiotic resistance. Antibiotic resistance genes example penicillin resistance. It means even in the presence of antibiotics like penicillin, they survive, grow and multiply, while the all other bacteria die. Selected PBR322 plasmid with antibiotic penicillin resistance are extracted from the bacterium and are cleaved by same restriction endonuclease EcoR1 that has already cleaved the desired DNA sequence, the insulin gene. These plasmids act as vectors that self-replicates and carries the ability to bind the desired sequence, the insulin gene. DNA ligase is then added to combine both the cleaved plasmid with the antibiotic resistance gene and the desired DNA sequence, the insulin gene, both of which combines to form recombinant DNA inside a hybrid plasmid. Now, self-replicating plasmid with antibiotic resistance has acquired desired DNA sequence, the insulin gene. Sometimes synthetic DNA fragments are added to bridge the ligated ends. These synthetic DNA fragments that bridge are called as linkers. Hybrid plasmid is then introduced into host bacterial cell E. coli. Process called transformation involves calcium chloride heat shock treatment. Incubating host cell and plasmid together at 0 degrees Celsius in calcium chloride solution and subjected to heat shock by rapid temperature shifts between 37 and 43 degrees Celsius. Following which the host bacterium E. coli acquires the vector plasmid PBR322. At times to clone longer DNA segments, BACs or YACs are used. BAC bacterial artificial chromosome insert size 100 to 350 kbs. YAC yeast artificial chromosome insert size 100 to 1000 kbs. Further, E. coli are grown in the culture medium that allows it to grow and multiply. The other unwanted bacterium that are grown during culture are killed by the addition of antibiotic, example penicillin. Host bacterium continues to grow and multiply as the plasmids provide antibiotic resistance to the host bacterium. Host bacterial growth and multiplication indirectly leads to multiplication of desired DNA sequence, the insulin gene. They are then pipetted or looped to special transfer tubes and are subjected to lysis through mechanical or chemical means, followed by isolation of hybrid plasmids. Hybrid plasmids are then subjected to cleavage by the same restriction endonuclease ECO-R1, which yields multiple copies of desired DNA sequence, the insulin gene. If expression vectors are used during cloning, insulin gene will be expressed to form recombinant insulin that can be isolated and purified to be supplied as insulin injections used for treatment of diabetic patients. To summarize, rDNA technology involves 7 steps. Step 1, extraction of DNA followed by cleavage of desired DNA sequence. Step 2, plasmid isolation followed by cleavage. Step 3, ligation of cleaved plasmid with desired DNA sequence. Step 4, introduction of hybrid plasmid into host bacterium by transformation. Step 5, selective multiplication of host bacterium in culture medium. And step 6, lysis of bacterium followed by plasmid isolation. Step 7, cleavage of plasmids to get desired DNA sequence that are amplified. This is recombinant DNA technology. Knowledge is beauty. Let your beauty progress all life. Signing off for now.